is to increase your understanding of the concept of synthetic lethality and help you can use the code to identify synthetic lethal interactions. So first, I'm going to continue with a few slides which were left from our class session. So in this session, I introduced that we can use the concept to identify new drug targets by identifying the synthetic lethal partners of cancer drivers. And here I want to also tell you that how we can use the concept to identify those patients who will have more response to a given cancer treatment. So here is the idea. So we analyzed the scope of the trial. So we performed a retrospective analysis. So we analyzed this clinical trial data for transcriptomics data. So first we compared, even if the synthetic lethality score of the different drugs, even if, so that would be our top recommendation if they provide the patient's data to us. So overall, based on our metric, then we could recommend the drugs that could be effective for the patients for 54% of the patients. And based on the winter trial paper, that they say that 26 are the result of our analysis, and everything, all the treatment options, including those treatments that she had received, didn't reach the criteria for our recommendation. So our recommendation already predicted that those drugs won't work. But our analysis showed that the patients would respond to this anti-PD-1 therapy, which is a powerful therapy. And even if she has, and this is the CT after 6 months. So her tumor reduced dramatically, and she is staying in complete response as of today. So this example was the great multi-analysis about the clinical trials of synthetic lethality. But I'm gonna skip this. The summary of this work is that the number of clinical trials and preclinical studies are increasing to identify new targets for cancer based on the concept of synthetic lethality. And what is more interesting is the non-synthetic lethality oncology trials or non-oncology trials. I think this is because synthetic lethality concept, then it has. So, GitHub link is not working. Do you have any problems? Let me know now. And for those who are online, if you have any problems in downloading the data or code from the link that we provided, please let me know. To their respective single knockouts, there are multiple sources of that kind of in vitro experiments, but it is very sparse, and it is not focused on certain cancer type, or it is not focused on certain therapy. There is certain molecular pathway, so it is very much scattered, and it is difficult to construct one single framework to predict the synthetic lethal interactions. And what is a more fundamental problem is that all these experiments were done in vitro cell lines. So even if we have good accuracy in predicting these synthetic lethal interactions in vitro, the resulting synthetic lethal interactions may not be directly applicable for patients' treatment. But our aim was to use the concept of synthetic lethality to treat cancer patients. and we wanted to identify what are the synthetic lethal interactions that are just like BRCA-PARP. So what will be the next BRCA-PARP? That was our question. So instead of focusing on those synthetic lethal interactions that are identified in cell lines and trying to predict them, we kind of took a different approach here. So we initially collected all the synthetic lethal interactions that are identified in vitro. So we manually curated we mined the literature, we performed the literature search, and we identified like 20 or 30 different papers and published it over the last 10, 20 years. And we manually collected all the synthetic lethal pairs that have been identified in these studies. And of course, we did not look at those studies where they studied specific pairs, like mutation of one gene and the other gene, and their co-mutations. And then if the co-mutation is less than expected, then we can kind of think that this will be strong evidence that this could be the synthetic lethal interaction. Of course, there can be other reasons that this phenomenon can happen, but we are using that as one of the evidence that it could be synthetic lethal. And the second idea is that if gene A and B are truly synthetic lethal, then those patients where these two genes are co-mutated will have better face. And also, like we perform this phylogenetic screening, by which we try to identify those gene pairs that are functionally related. Okay? For example, the best known synthetic lethal partner, BRCA-PARP, both of them involves DNA damage response pathway, right? So BRCA is double strand break, fixes double strand break, and PARP fixes the single strand break, and co-inactivation of them leads to cell death. So these two genes has to be functionally related. So for that purpose, we use the phylogenetic profile and selected those genes that have a similar evolutionary history. The reason why we performed this screening was to remove false positives from our first and second screening and to increase the chance to get the true synthetic lethal interactions. Okay, so this is our method. So we started from the cell line identified synthetic lethal interaction, which has some experimental evidence that they are synthetic, that almost none of them passed our screening about the patient's relevance. So we collected about 3,000 synthetic pairs that are experimentally identified, but only one. So we changed our strategy. So instead of looking at the double knockout or isogenic versus genome-wide knockout data sets, we looked at the large-scale cancer cell line data, which is called GET-MAP, cancer cell dependency map, and we used the gene expression, copy number, or mutation data, and their gene essentiality data, which was obtained by RNA interference or CRISPR knockout. Okay, so if you go to, I'm not sure if you have experience with the GET-MAP data, they have performed genome-wide CRISPR screens or genome-wide RNAi screens for the entire all protein coding genes for about 600, 700 cancer cell lines. So we overlay that with the expression, copy number, or mutation data, and to identify the synthetic lethal partners. So what do I mean by that? So let's say if you want to identify if you want to check whether gene A and B, they are synthetic lethal, then you have cell lines from the cancer cell lines. You have cell lines where gene A is mutated and gene A is wild type. So from the collection of 600 cancer cell lines, you have those genes where gene A is mutated and where gene A is wild type, and compare the essentiality of B measured by either CRISPR or RNA interference. Okay, so if they are truly synthetic lethal, then the knockout effect of gene B will be more detrimental for those cell lines where A is mutated. Because when A is knocked out, when A is mutated, genetically relevant synthetic lethal partners. So I'm sure you have already downloaded the code from this webpage, and you have the data that you need to run this code. Okay. And this is the first step of the inference of synthetic lethality. So like I said, we collected all the double knockout results and single isogenic cell line experiments. And also, we looked at the DMAP data. We analyzed the larger scale single gene knockout screens with wild-type transcriptomics. SL equals C bind 1,000 genes, and we are checking the potential synthetic lethality between these three gene pairs. Okay? And before you run this, you have to R2C interface. On server, it's a Linux server. But and I'm also gonna give you if there is any way to solve this problem in Mac, I'm gonna give that to you. But it should be possible. But I'm sure that the department has the server that are open to every one of you, so there'll be no problem in using it. So I'm gonna show you what this code can do for the rest. Are using Windows. Inactivated, gene B is more essential. And this is the very first step where we collected the golden set of synthetic lethality. We looked at the literature and mined like 20, 30 different papers. And then we identified all the pairs that are performed, pairs obtained from the double knockout experiments. But these are mostly not relevant. So, because none of them passes the clinical relevance test. And this is the test where we identify those pairs that pass the cell line screening. Okay. And then let me go to step 1. Here we load the TCGA data. So here we start the screening in the patients. And so for the first pair, yes, it is less than expected. The p-value is 0.0003. But for the second pair or third pair, it is not less than expected. So our p-values, they are the coefficients for the hazard ratio. This hazard ratio means if they are positive, then means that aggravates the patient's prognosis, and if they are negative, then they like kind of enhancing the patient's prognosis. And these are the p-values associated with that. So actually all these three synthetic lethal interactions, their co-inactivation never improves the patient prognosis, but they all decreases the patient prognosis. So our analysis shows that all three pairs are not synthetic lethal interactions that are clinically relevant. Here, yeah, you're right. So these are the coefficients, and these are the p-values associated with that. So actually, the first pair shows the negative coefficient. So, and so this is the last the results from our last screening. So this PP2 means the phylogenetic profile. Their closeness in the phylogenetic profile. So the smaller the value, then they are close to each other. The larger the value, they are far away from each other. So actually only, and we are using the criteria of 0.5 here because that, based on our analysis, that is the criteria where we, in our test set, that is the criteria that can best distinguish synthetic lethal versus the positive versus negative set. So here in our fourth screenings, but if you want to, depending on your laptop, this function may not work. So in this code that I'm showing you, I have removed those options to use the multiple cores. And then there's one more thing you have to change. The feature map has to be defined as, and other than that, you won't have any problems. So that has to be fixed. Okay, so today we have covered the concept of synthetic lethality and how we can use it to advance precision cancer medicine. And we have gone through the code step by step, and I have provided you all the data and the code to run the synthetic lethality pipeline. So if you have any questions in running it yourself, please let us know. And I'm gonna also check how you can set up a GCC compiler properly in MacBook, and I'm gonna let you know. I'm gonna email you through for all the participants.